Good afternoon, everyone. First of all, I would like to thank the organizers for the invitation. In principle, I didn't know how my work would fit in this quantum biology field, but watching the presentations here, I started to understand how it can contribute to the growth of this field. I'll be talking about our recent work on cell viscoelasticity as a biological marker of the cytoskeleton, the structure, and diseases. But first, I work in Universidade Federal do Ceará in the Laboratório de Física Biológica, and we are mostly interested so far on the physics of living cells and tissues, the mechanical properties of soft materials, and theoretical and computational methods in biophysics. So my presentation is organized. My presentation is organized as follows. I'll briefly talk about a little bit on cell mechanics. I'll be very quick on the basic principles of atomic force microscopy. I don't know if the audience, if most of you know this technique. I will review some work, my recent work on cell viscoelasticity as a biomarker for diseases, and how some recent results, very in the beginning of our research, of how the cells are interacting with light. So this is, most of you know this better than I do, is that how cells are complex, especially for a physicist as myself. And the way I see that we can see two different compartments. There is the cytoskeleton, who gives structure and rigidity of the cell, and the organelles, who gives the function. But together, they perform all the complex functions of cells, like cells move. This is a time lapse of a wound healing experiment. Cells, they also invade tissues. And we can ask, are cells solid or fluid? They look like, cells move like Venom, that enemy of Spider-Man, right? I like this, the way the cells move. So actually, cells are viscoelastic objects. And to give you some numbers of the elasticity modulus of cells, they range here around 1 to 10 kilopascal. This is the Young modulus of the cells, and compared to other components of the human body, like bone and collagen, the cells are like orders of magnitude softer than these other harder, these most stiff parts, like bones and collagen. And the way we probe the viscoelasticity of cells is with atomic force microscope. Essentially, we make images. The atomic force microscope work like the same way that someone, a blind person, reads those texts. I don't know, Braille, I don't know how to say that in English. Okay, so I will briefly review how the technique works. The AFM, it consists of a flexible chip with a reflexive coat here. There is a laser pointing to the back of this tip, and the way it interacts with the surface will make it bend upwards and downward, and this movement is captured by a photodetector sensor here, and then you can construct the topography of the surface. And this sample, it moves in the X, Y, and Z, so you can construct the topography, as I said, but you can also apply tiny forces in a very controlled way to the samples. I'll skip this one because it's just to say that the way these forces are detected, we approach the deflection of this flexible tip to Hooke's law, the physics one in the curriculum. So what we do here to probe ourselves is that we touch the cell. We move the Z scanner, and then it touches the cell up and deflects up to a maximum force, and we construct these force curves here. And using some deformation model, we can extract all the viscoelastic properties of the cells. And to measure cells, this is our workflow. Essentially, we take the whole cell culture to the FM, and then we scan the surface of the cells, and for each point, we acquire a force curve. Then we make several force curves in the whole cell surface. We apply a physical model, we transform it to a viscoelastic map, and then we make statistics in several measurements in different cells. The first story I want to tell is our first paper. I'm originally a theoretician, and I became an experimentalist about a decade ago. We took 3 cell lines from kidney. There is one normal cell line and 2 different cancer types. There was, I would say, a less aggressive cancer. This is the cell line, and a metastatic cancer. And what we observed is that cancer cells are softer and less viscous than normal cells. And the more aggressive is the kind of cancer, the softer the cells are and the less viscous they are. And this is what we obtained here with the FM. But then we needed, this is just, there was no biological meaning in these numbers. So we performed this regular, this is from the people who biomedical sciences. This is regular experiments they do, the confluence, how the cells divide, cell motility. And this is a very qualitative analysis of the citoskeleton disorder. It was by eye, with a specialist look at the cells and said, this is more organized and this is more disorganized. And what we observed is that this nanomechanical analysis presented a strong correlation with what was observed with this biomedical application. So it was for us a very convincing result that yes, the cell mechanics can be used as a biomarker for cancer cells, or for the aggressive of cancer, and can also be used to other types of diseases. But cells are very complicated viscoelastic objects. Several works in the literature says that there are two viscoelastic regimes. There is a fast relaxation regime that occurs for time scales below 10 milliseconds, and there is a slow viscoelastic regime that for time scales larger than 10 milliseconds. And also cells, they relax following a power law. And then we went back to our model and we developed a 2 power law viscoelastic regime. And now instead of only one number, like the Young modulus to describe the elasticity of cells, now we have 4 parameters. We have the elasticity modulus, we have 2 exponents. Those exponents are directly related to the health state of the cell, and also to the structure of the cytoskeleton, and this time scale that is related to the time scale that divides what is the fast relaxation to the slow relaxation regime. And we knocked on the door of our collaborators, and we get as many cells we could find. And then we mapped the viscoelasticity of them with these 4 parameters. And if you look, these are the 2 exponents. The lower one are the slow relaxation exponent, and the green one is the fast relaxation exponent. If you look around, if you look at this graph, those numbers, they vary a lot. Of course, we took cells from different kidneys, from different human organs, and with different stages of the cancer, and they perform different functions. Of course, they have to be different somehow. So we are also after some biological meaning to these numbers, and we performed some experiments. Again, we performed the motility and invasiveness experiments in a special group of those 13 cell lines. We took gastric cells, and here again we have a normal line, less aggressive line. We have a metastatic cancer and an invasive cancer. In principle, I asked to my collaborators, what is invasive cancer that is not metastatic? And they explained to me, well, you know, when you have a tumor that you don't have a very specific shape, it's kind of diffuse. It's because you cannot see where the borders are, and they are diffusing towards another tissue, and you cannot say where, you cannot extracting easily. So they say that it's a very difficult cancer to fight against. These are cells from patients. And what we found is that, I'll change slides here, is that, again, we found a strong correlation between the biological functions and the mechanical properties of those cells. The motility is the inverse. The most aggressive type of cancer was the cells that exhibit the largest motility properties. The normal cell was the less motile, let's say, just to speak like that, cells. The crossover time is that time between that separates the slow relaxation to the fast relaxation. It has an inverse relationship with the motility. The invasiveness correlated very well with the inverse of the elasticity modulus. The softest kind of cell was the invasive one. The most rigid cell was the normal one. And this is for the gastric cell group. But we also had some in those cells. We also had the group of kidney cells. We did not perform all these experiments, but the motility, we were able to find the same cells, a motility study of the same cells. And again, we found perfect correlation between mechanical properties and the aggressive of the cancer. But now, under the light of a time-dependent relaxation of the mechanical properties. So in that paper, we proposed a model saying like this. This is a double power law relaxation function. And the fast relaxation regime that exhibits a larger exponent is related to the long filaments of the F-actin cytoskeleton that they at room temperature, they vibrate. So those vibrations of this large number of filaments contribute to the fast relaxation or viscoelastic relaxation of the cell. So it's not related to the cell function at all. The regime that's related to the biological functions, that is slow relaxation, that is related to the mechanical deformations and remodeling of the cytoskeleton. And also the crossing time between the fast and slow relaxation regime, which we believe that it's some sort of minimum time scale for cells to perform their biological functions. So the smaller are this crossing time, the more the cells exhibit larger motility. This is what we observed in our experiment. So in the light of these observations that the cells, that the viscoelasticity of the cells are somehow connected to the biological functions and motility mostly as well. We propose that cell viscoelasticity is a powerful lens to investigate biological phenomena in cells. Even without, we can see from just pressing the cells, what's going on inside them. Just a quick story. We also, I talked about the structure in my presentation. We also put those cells on top of soft gels. The first set of experiments was in top of glass in the Petri dish, and the cells responded completely differently. In our experiments, what we observed, the cells over soft gels, not so soft actually, this is the range of rigidity of the gels, that those cells are stiffer than the cells put on top of the glass. And we didn't know how to explain that. And what we did is we observed. We also perform dynamic measurements. We put some frequency on the force that we put on top of the cells, so we could separate the elastic response to the viscous response. And the largest contribution was in the elastic response, which suggested us to look what's going on to the cytoskeleton of the cells. So we took the confocal images of those cells and tried to perform a more quantitative analysis. What we did was we calculate the fraction, the fractal dimension of those images. And what we observed is the fractal dimension increased as we increased this is the rigidity of the substrate. This is the very soft gel. This is also gel, but 30 times stiffer than this one. This is glass with and without collagen on the surface. And comparing what is going on with the structure of the cytoskeleton with the mechanical properties, at least in these cells, this is our fibroblasts, is that the rigidity scales inversely with the fractal dimension, but the fluidity of the cells increased. Our data suggests that cells on top of glass, on top of very hard substrates, becomes more fluid than the cells on top of very soft substrates. I cannot explain, I'm not a biologist to explain what's going on, but probably the cells are feeling the soft gels and trying to indent it, try to invade it. I don't know. That's what we are still thinking about this. It's a manuscript in preparation. We don't have all the answers so far. So what we conclude on this work is that the viscoelastic properties and cytoskeleton organization are affected by the stiffness of the extracellular matrix. So, sorry. And so you can modulate the properties of cells, the viscoelastic properties of cells with diseases can do that. The extracellular matrix the rigidity of the extracellular matrix can do that. And then when I found Marcelo a year ago, I found out about his company, the Bright Photomedicine, and I wrote him and said, can you give me your equipment so I can shine some light on my cells to see if the light can modulate the viscoelastic properties of those cells? And it was a year ago, about not that much, but let's say it's 1 year, that I got I knew about photobiomodulation. And looking at the references, I found this one that they realized that the cells, the motility are modified when you put some this is two different wavelengths. And you see here the motility when you have a control, no light, and then we change the power of the lights were subjected to with two different wavelengths. Sorry. And you can see here that the light is affecting the motility very clearly. Two different wavelengths, one better than the other, but the light is affecting the motility. And remember that motility is encoded on the viscoelastic properties. Also, light is affecting the structure of the citoskeleton. In this work, they made some image analysis of different aspects of or different characteristics of the citoskeleton. And they realized that light is also changing somehow the citoskeleton, the F-actin citoskeleton, the cell area, and the number of filaments, and even the total response, optical response of the F-actin. And what we started to do is that we took two different cell lines. We took fibroblasts and osteoblasts, and we exposed it to light, to bright photomedicine equipment, and we observed that the fibroblasts just don't care about the light at all. However, the osteoblasts, they respond very, very convincingly. Well, here we only analyze the cell viability to only 50 milliwatts per centimeter square of light for only 20 minutes, which is okay. So my collaborator will prepare these cell lines. He's told me this. He told me that he tripped over this subject again. He studied something related to this many years ago, and he said that, well, this is data very important for dentists, because they say that when you remove a teeth you are planning to put an implant, you have to do right after you remove it, otherwise the tissue will invade the bones, and it'll be very difficult to maintain the implant. So this shows that you can avoid the proliferation of the tissue, the gingiva, I don't know, the gums, right? Towards to the hole. If you expose to light, you can stimulate the growth of fibroblasts, and then you have bones, so you can hold the implant. So our next steps, these are experiments that are running. We don't have more data because our AFM is broken because of the pandemics. Too much time exposed to humidity, and we are making now motility and wound healing experiments, proteomics, and also this is the nanomechanics, the last part. But our expectation is viscoelasticity modulated by light? If in the literature says if motility changes, the light changes the motility, I'm pretty sure that this crossing time and these exponents will be affected by light. And I'm sorry, I expired the time for a few minutes. So take-home messages are: cell viscoelasticity is a powerful lens to investigate biological phenomena. The viscoelasticity of single cells are strongly correlated with the aggressiveness of cancer. Cells exhibit a power law-like relaxation function, and the relaxation function encodes some motility trends of those cells, and that the AFM is a very powerful tool to investigate those cells, because we can probe them while they are alive, and we can make intervention, pharmacological intervention, as we measure those cells. I had some extra slides to show, but then I expired my time. I'm sorry for that. So I thank you very much for your attention.